Welcome to Faculty Focus, the podcast of the Faculty of Pharmaceutical, Biomedical and Veterinary Sciences of the University of Antwerp. Live from our annual Faculty Research Day, where we highlight the research of our professors and PhD researchers. I am Teun.
And I am Marleen.
Hi. Our first guest on the podcast today is Professor Daniele Bertoglio. He works as an assistant professor for the Department of Biomedical Sciences at the Bioimaging Lab. His research focuses on the application of state-of-the-art small animal neuroimaging tools to quantify structural, functional and molecular changes in preclinical models of neurological diseases. In his presentation today, he talks about how in vivo imaging advances our understanding of neurological disorders. Welcome, Professor. Let's maybe kick off with a very broad question. Why is studying the brain still such a challenge compared to other organs?
All right. First, thank you for having me. It's my pleasure to be here. So studying the brain is difficult. One of the main reasons why we have troubles or challenges with looking into the brain is its inaccessibility. Because unlike other organs or parts of the body that we can easily collect a biopsy or look at the tissue in different ways with a surgery, reaching the brain is very invasive, requires very complicated neurosurgeries, and therefore most of the time we cannot do that, especially in the early stage of a pathology.
Okay.
So that's why you also use neuroimaging, right? Yeah.
Yes.
Yeah. What can neuroimaging reveal when we look inside the brain? What kind of changes or patterns can we actually see?
So with the power of neuroimaging is the fact that, first of all, it's non-invasive. So we can look at the brain, the different parts of the brain without having to intervene on the subject. And it allows us to see the whole picture of the whole organ together, meaning that also deeper brain structures that are very difficult to access otherwise can be monitored. And with neuroimaging, we have a very wide variety of tools we can use to look at the structure, but also at the function of the brain, so that we can really understand whether specific alterations in a pathology are more related to, for example, volumetric changes of a brain region, or if it's more functional, and therefore the way different brain structures interact among each other.
Okay, let's talk about those techniques. There are two techniques you use a lot in your research: the magnetic resonance imaging, MRI, and the positron emission tomography of PET. We know the abbreviations, but can you briefly explain the two imaging techniques to us?
Of course. So with MRI, we use actually as the word says, magnetic resonance imaging. So it's basically a very big magnet that we use. The machine is a big magnet, and we utilize the resonance features of the water molecules that we have in our tissue, particularly the hydrogens. And with that, we can recreate a three-dimensional image of our body. While with PET, with positron emission tomography, we are injecting in the patient, in the subject, radioactive molecule, which releases positrons, and we are able to detect with the crystals of the machine. And by looking at that, we can understand where the radioactivity distributes in the body.
I know they can't hear you, but do you have a favorite?
I like them both.
Okay.
I don't.
No favorites on the table.
No, they are very complementary in the sense they provide extremely different informations, and where the MRI is extremely powerful in terms of structural and functional informations. The PET imaging provides us key information from the molecular level. So they have both of them some advantages and disadvantages, and the main power lies in the multimodal approach where you combine them both.
Okay, and you focus on Huntington's disease. Do we already fully understand how the Huntington's disease work? And what causes this illness?
So from the pathophysiological perspective, we have a very good knowledge about it because it's a monogenic disorder, dominant disorder, meaning that there is a single mutation in one gene that causes the pathology. And the gene is the Huntington gene. And when there is a specific part of this gene that is expanded, this causes the protein that is formed to be aggregation prone and resulting to toxic to the neurons. Now, although we know the cause of the pathology, understanding how we can cure the pathology, it's much more complicated, and we have been working in the field for about 30 years since the discovery of the gene where it is causative. And right now there are quite a number of clinical trials targeting the modulation of the mutant huntingtin protein to try to slow down the pathology.
Okay.
Right. And in which stage are those clinical trials? Do you know? Are there some almost finished?
So there are. So the very first trial trying to modulate mutant huntingtin started in 2019, and that was stopped a bit earlier due to some side effects that were observed in the patients. However, there are now more than a handful of trials that are in phase 2 or phase 3, and we are really looking at the first stages of safety assessment, and now we are looking at the efficacy of these treatments.
All right, so it sounds very promising.
It's although it's a rare disease, the pharmaceutical field is quite active, and there are several type of treatments looking at the pathology or targeting the pathology from different perspectives. So that makes it quite hopeful for the community that either few or some of them will actually work.
Yeah, which is not really common for uncommon diseases that pharmaceutical companies are working on it.
Yes, indeed.
Yeah.
It's true.
Yeah, and maybe a follow-up question on that. Can studying well-understood diseases help us learn more about those that aren't fully unraveled yet?
I believe so. Like in the case of Huntington disease, so the expansion in the gene is related to a specific sequence of DNA, so a CAG triplet repeater that it repeats a certain number of times. And this is the part that is prone to aggregates and cause the toxic, exert the toxic effects in the neurons. But there are also many other triplet repeat disorders that are present. They are caused by triplet expansion in completely different genes, and they lead to a completely different pathology of the central nervous system. And if we find very good or promising therapeutic approaches in the context of HD, it might very well be that some of these might be applicable or will help us to better develop treatments for these other disorders.
You use specific biomarkers, right? You describe them as safety biomarkers. What are they exactly, and why do we need them?
Our work in the past years has been focusing on identifying biomarkers that are helpful to monitor treatment efficacy or follow up the progression of the pathology, and those are usually more known as prognostic or predictive biomarkers. However, they don't necessarily inform us whether a specific therapeutic approach is safe or not, and that's where the safety biomarkers come in, because we need to have ways of making sure the treatments or the drugs that we are giving to the patients do not worsen the pathology, and they do not worsen the quality of their life, which is exactly against where we want to go when we are trying to give a therapy. And the importance of safety biomarkers in the context of Huntington disease became more relevant in the past years, because after the first couple of clinical trials got halted due to safety concerns, the community realized we are actually lacking ways to make sure that, we are lacking ways to monitor whether a treatment is having any adverse effect.
And your work is a direct bridge between experimental findings and clinical applications. Can you elaborate a little bit more about that?
Sure. In the lab, we work with small animal MRI machines, and these are based on the same physical principle and same technique, but it's the one from the clinical hospitals. Mm-hmm. This means that if we develop a biomarker, being for therapeutic efficacy or for safety evaluation, the type of biomarker that we develop can be immediately applied to the clinical setting.
Okay.
And in that sense, it's another particular key feature of neuroimaging is to be translational. So we can go from the bench, so from the preclinical work, to the bedside, to the patient.
Okay.
So if you want to do a translational research, are there other aspects where you have to focus on? We have to be aware or take more into account than normal research? Or not normal research, but more fundamental research.
Well, when we want to have our research to be translational, it's very important that we consider what are the final needs at the level of the patients or the human evaluation in general. So one aspect is, of course, the methodology, so the MRI, so the instrumentation that we use, but it has to be something that can go to the humans, being translational, but also another important aspect is the preclinical research that we do has to be translational as well. So we have to use animal models that are as close or as similar as possible to the pathology that is described or is observed in the patients.
Yeah, all right. And if you have translational research, you have like grown-ups, but also children. Is there a difference there?
We do. So the advantage of neuroimaging is that it's non-invasive. So there also we can utilize it to study longitudinally the evolution of a specific biological process. So we have been working actually in the past couple of years in collaboration with our groups, other groups here on campus at the VIB, on a pediatric form of epilepsy that we studied in mice. So we started scanning animals as of 2 weeks of age, and we follow them up over time to understand how their brain will develop and what will be different compared to the normal development.
Okay. And so when you use MRIs or PET scans, because it's non-invasive, I can imagine that you go to the clinical stage much faster than when it's invasive?
There is the possibility,
yeah.
Yes. So the advantage indeed is that we can, whatever we found that is relevant to be brought to the clinic, we can immediately reach out to the clinicians and to the radiologists so that they can try to implement it at the clinical level, yes.
Okay.
So you're going to present this afternoon at the Research Day. Is there still something you'd like to add to this podcast that you think the listeners should know this?
Now we have been talking about Huntington disease, but in our group we work also on other pathologies, of course. Because we want to understand in general the entire central nervous system. So in that regard, we also develop biomarkers for other pathologies, like more known, for example, Alzheimer's disease, as well as multiple sclerosis. And since I joined the lab a couple of years ago, we also started a research line on spinal cord injury, where we utilize animal models of contusion spinal cord injury to understand how we can track and monitor the changes at the level of the spine, but also what happens in the brain itself as a consequence of the spinal cord injury. Because following the injury, there is you have limited mobility of some of your limbs, and this will impact the way your brain works and will cause some brain plasticity.
Okay.
And this plasticity is what we are trying now to visualize to potentially help developing better treatments, focusing, for example, on brain stimulation.
Okay, like against phantom pains.
Yes, exactly.
Okay. Yeah, I think we can wrap up this episode. Thank you very much, Professor. And maybe to finish, what advice would you give to a young aspiring scientist?
What advice? I think the main advice will be to be curious and always try to understand what you're studying, what you're studying, what actually means. Not taking the notions that we know from the biology, let's say, for granted, because we keep learning new things over time.
Okay,
but that's very relevant because in the next podcast episodes we will breach through some dogmas in the research. So, thank you.
Thank you very much.
Okay, thank you.
Our next guest on the podcast is researcher Rita de Souza. Welcome to the podcast, Rita.
Thank you.
She works at the Department of Veterinary Sciences at the Campus Research Center on the production of follicles from ogonial stem cells and calls this the long-awaited solution for fertility preservation and restoration. So, in the introduction to your research, you state that more than 80 studies challenge the dogma that females are born with a limited number of oocytes and that ovaries lose their capacity for oocyte renewal at the time of birth. Is this the result of a male-dominated science landscape, you think?
Okay, starting very controversially, I think.
It was your introduction, not mine.
Yes. I wouldn't. Yeah, the scientific world was indeed male dominated, but I think there's also female scientists that keep on pushing that this dogma is strict and fixed and it will never change. And I think that's not a way to approach science. You should always be open-minded to discover new things. And that's why my research always keeps on fighting and fighting and fighting.
So it's a bit like the Earth is flat and the Earth is round type of question, right?
Yeah.
So is the long-held belief that women are born with a fixed number of egg cells now really being challenged?
Yes. Ever since 2004, there have been so many research studies challenging this dogma, and there has been offspring produced in mice already from my ogonial stem cells. So, yeah, you keep on giving evidence that this should and can be challenged.
So what could this mean for the infertility treatments?
Yeah, it means a completely change of way of thinking. Anyone that, well, imagine if you're a menopausal woman, you never had the chance to have your own kids, and now you want it for any other reason. Then the ogonial stem cells brings a way that you can't do it like that. Or, for example, if you are a prepubertal girl and you had cancer or something, and your oocytes were compromised by it, you don't have oocytes anymore, then that's a way that you can still be a woman and have your own kids. If men can have spermatogonial stem cells and have kids until they are 90 or more, women should be able to also. So, yeah.
And maybe you have to define the ogonial stem cells for us.
Yeah, so it's a term for germ stem cells that are found in the ovaries, and they are already there. So a stem cell should and can be differentiated into something else. And these ogonial stem cells, they have the ability to differentiate into new oocytes.
Okay, so. And you talk about pre-existing ogonial stem cells. What can you elaborate on that?
So pre-existing means that they are already there. So I'm trying to say that the ogonial stem cells already exist in the ovaries from the day we are born. That's different from the induced pluripotent stem cells, which is something we create in a lab.
Okay. And how do you identify these cells?
Yeah, it's called with a technique called fluorescent activated cell sorting, where we sort for a very specific germ cell marker called DDX4. And then the oogonial stem cells, they are extracellular positive for this marker, whereas all the other germ cells that are in the ovary will not be stained for this.
Okay.
And then we can really separate our population from everything else that's in the ovaries.
Okay.
Do you know where in the ovary it's resided?
Really. We still, that's one of the controversial topics where they actually are. Some people say they are more in the ovarium epithelium surface, but they can be anywhere in the ovary. That's something that we still need to discuss and research.
Okay. You also use ovine 3D organoids.
Yeah.
What are those exactly?
Yeah, so it's a very fancy way of saying I couple cells together and I form like a kind of 3D structure, like a ball with a mix of cells. And I use my oogonial stem cells and other type of somatic cells that are already present in the ovary, and I bring them together, and then I make kind of a ball of cells.
And do they just form a ball together, or do you have to add other factors
no,
for
we
it?
use a centrifuge, and then the cells couple together, and then they are in specific plates, and then they just make a ball.
Okay. Make it sound so easy.
Yeah.
And how big is that ball? Do you have an idea?
It's really, really small.
So nothing olive-like, it's just more...
Yeah.
It's smaller than that.
Yes, it's very small. You can see it in the microscope clearly, but not
Not with the naked eye.
Yeah.
And your research, or the research around this, started in mice, but you focus on large animal models.
Yes.
Yeah. Why?
Yeah, we started with a pig to bring it closer to human research. So pig is very more similar to humans than mice is. And there's a very less scientific way to say that everything works in mice. So we wanted to prove that we can show it in larger animal models. And we also wanted to look not just into human research, but also endangered species research. So we wanted to be as broad as possible. And with a larger animal model, you can bring the research closer to these kind of human and endangered species. But we also now, we do pig, we do cow, we do sheep. So as many as possible, basically.
The llamas here as well?
No, not the llamas. Yet.
Yes. So is this an invitation?
Yeah, well, the thing is, we always need to wait for an animal to die. So for the pig and the cow, we get it from the slaughterhouse and also the sheep. But if an animal dies at a zoo or something, there's always a possibility that maybe we can get an ovary and then we can do some tests.
Okay.
But it's a matter of waiting.
Because we already, on the podcast, we already had Professor Ruud Appeltand.
Yes, she's my promoter.
Yeah, this is your promoter. And how does your research fit in her work with in vitro gametogenesis?
Yeah, so basically what I do is in vitro gametogenesis, but her real true passion is indeed about the rhinoceros. Yeah. And so pig is a bit far away still from the rhinoceros, but we do have tapir ovaries that maybe eventually I can also use and get oogonial stem cells and then produce new follicles, and then she will be very excited.
Okay.
Is there something we didn't ask you about your research and the talk you will give during the research day?
No, I just hope everyone likes the talk and that it's clear enough to understand. It's a very new topic, and I hope everyone can be open-minded about my work.
Okay. And maybe we want to finish off with a broader question. What advice would you give to a starting PhD researcher?
Yeah, be realistic. You need to have a lot of passion for the work you're doing, more than any kind of normal job, because you will struggle. That's the reality. You always imagine, it's going to get great. In my case, I would produce like a mini pig from the oogonial stem cells. It will be amazing. But that's never going to happen. That's not going to happen while I'm probably alive. Yep. So you need to tone it down, have a passion, and aim for like the world, but be realistic that it will probably not gonna succeed.
Okay.
Yeah. And
just take it step by step.
Yeah, your research is just a piece of the puzzle.
Yes,
right? Yeah.
indeed.
And the whole puzzle is difficult to achieve in one PhD.
Yeah.
That's true.
Okay. That's good advice, I think. Thank you very much, and good luck with your presentation this afternoon.
Thank you.
Thank you.
Next on our Faculty Focus podcast table, we have PhD researcher Paula Polanco Miquel. She works at the Microglia and Inflammation in Neurological Disorders Lab at the VIB Center for Molecular Neurology. Her research presentation today is titled Lysosomal Failure Triggers Microglial Degeneration and Accelerates Neurodegenerative Pathology. Hi, Paula.
Hello. It's not an easy title.
No, it's not an easy title. And that's not an easy subject as well. But before we take a closer look at your research, can you tell us what those microglia are? What do they do exactly?
Sure. So these cells are the sort of like the guardians of your brain, and you can understand them as the immune system of the brain. So basically, they take care that everything that is going on in the brain, it's flowing smoothly. So they are, for example, very important in any sort of disease context.
Okay. And so that's why they are important, I can imagine.
Exactly.
Okay.
Yeah.
Okay.
Especially like in the context of neurodegenerative disorders, where there is like a disease ongoing. These are the cells that will take care of the neurons and that everything that is going on around.
Okay.
And they also rely heavily on their endolysomal system.
Exactly.
This is also called the recycling center of a cell. Why do we need cleaners in our body?
So first off, indeed, we have this organelle within the cell that is called the lysosome. And as you said, the lysosome basically, you can understand it as the stomach of the cell. So in many pathological conditions, there needs to be a lot of clearance of the surroundings, and especially in neurodegeneration, because there is neuronal and cell death occurring, these cells need to be cleared up. And apart from that, in these diseases, there is also protein accumulation, sometimes extracellularly, sometimes within the cell. So the protein that is accumulated also needs to be cleared up. And basically, the microglia cells try to help with this. Doesn't always go well, but...
Is there also something as too much clearance? Too much clearance?
Yeah. Well, so this is like a discussion currently in the field, because indeed, on one hand, you want the microglia to be okay and like helping and cleaning up among the many other functions that they have, but talking specifically about this. But then there's also the discussion that sometimes these cells can get overactivated, we say, and these cells can enter like this feedback loop where it can become detrimental. So they can become too activated and maybe even phagocytose. So that basically means like engulfing something. Like it can signal for phagocytosing cells that should not be like engulfed, for example. So that's kind of what like my perspective is, that this needs to be really tightly regulated. So we need to be careful on how much we push these cells for like their activity.
Right. And an important lysosomal protein is progranulin.
Yeah.
Yeah. Where does it come from, and what does it do?
Yeah. So progranulin is it's a lysosomal protein. So indeed, it plays a key role within the lysosomes, and it is interesting because there are more than 70 loss of function mutations that have been identified within this gene, which basically leads to reduction of the protein. Sometimes it's 50%, sometimes it's 100%, sometimes it's even like very small deletions that have predicted loss of function. The general notion is that it has inflammatory activity. So basically, it's understood as a modulator of inflammation. And it's interesting to study this within microglia because, as I said, they are the immune system of the brain. And when you look at the expression levels of progranulin within the brain, microglia is the cell type that expresses progranulin the most. So that's why I put those two things together.
Okay. And what happens if progranulin doesn't function properly?
So what the most let's say, common characteristic is lysosomal abnormalities. Especially what I have seen particularly is that this leads to problems in the degradative system of the cell. So basically, it will, on one hand, for microglia, it will activate them a little bit more. These cells seem to be having more phagocytic activity, but the problem is that they eat up more, but then they cannot degrade it. So that's the feedback loop that can push the cells towards cell death.
They stay full.
Yeah, exactly. It's like they cannot degrade it. And then what I particularly saw is that because they cannot cope with this, then they will degenerate as well. I think in neurodegeneration, for a very long time, the field has focused a lot on the neurons, and in neurons dying, but nobody really questioned, okay, what about the other cells? Do these cells also die? So that's why we took it towards this side.
And for your research, you use a xenograft model?
Yeah.
Can you explain this to a non-scientist like me?
Yeah, basically, xenotransplantation or xenografting means taking cells, tissue, or organs from one species and putting it into other species. And in my case, we xenotransplant human microglia into the mouse brain to look at how they behave.
Okay. There is a link with amyloid beta plaques that are one of the main hallmarks of Alzheimer's disease. Can you explain a little bit how that is linked?
Yeah. Maybe I can start by saying progranulin is mainly associated with frontotemporal dementia. It's another type of dementia that leads more to behavioral abnormalities, like changes in personality. But as I said, there are also small mutations that have predicted loss of function. So it won't be like a 50% reduction of the protein, but probably you lose a little bit. And this is associated with an increased risk for developing Alzheimer's disease. Now, in my research, I worked also with granulin deficiency in microglia in the context of Alzheimer's disease, because I want to study how these cells react under neuropathological conditions. And AD mouse models are a great model for this, because you have A beta plaque accumulation, which is let's say a pretty extreme model of neuropathology.
When preparing this podcast, we come across a lot of mice research. Mice have taught us a lot in science, right? Do we appreciate their contribution enough?
Right, that is a good question.
Because
I mean, definitely, as a researcher, I can tell you we really do.
Yes.
And when we work with mouse models, there is a lot going on behind the scenes and a lot of regulations and a lot of certifications that we need to first acquire to make sure that, we give these animals the best possible life. So indeed, they have been huge contributors to this, and especially for neurodegenerative or just the neuroscience field, the brain is a pretty difficult organ to study, right? Yeah. So in this case, I would say we use them when there's just simply no other way to look at it better,
Yep. Back to your research, that microglial fragmentation and degeneration haven't been described before in the context of neurodegeneration. Mm-hmm. What could this new finding lead to, and why is it so significant?
Well, it's because, as I was saying, the field has been focused, the neurodegenerative field has been focusing a lot in the neurons. And I think over the past decade, there has been a bit of a shift towards looking at how other cells in the brain behave and how they are. And I feel bad for the microglia, because they are the ones that are helping and that are trying to combat these diseases. And what I saw once we started paying attention to this microglia is that they can also die, right? And I'm not saying they are more or less important than neurons. I think everything is equally important, but we need these cells to stay where they are, to be able to help the neurons.
And can they also be a part of the disease itself?
Yeah, absolutely. So I think nowadays, when you talk about neurodegeneration, you don't only talk about proteinopathy, which is like the classical hallmark of these diseases, is that there are proteins mislocalizing and accumulating in places that they should not be. But now they also, if you will ask somebody to define it, they will also describe that there is like an inflammatory response going on, which is the microglia basically responding to these insults.
And there are also solutions to restore the function of microglia. For instance, the pharmacological reacidification of lysosomes. What do we have to expect from that?
Yeah, I mean, because of what I see, particularly, I know I work with progranulin deficiency microglia, right? But at the end, what I'm looking at is how does microglia that have defects in their lysosomal system work, right? And what I saw is that the lysosomes of these progranulin deficient microglia, they are deacidified. And it's the same as your stomach. You do need an acidic environment to be able to degrade things. So that's why I used an acidifier to reacidify the lysosomes and look if we can revert these issues with degradation that we see. So indeed, when I tested those, I did see this phenotypic reversion where the blockage in the flux of the degradation was restored back to normal. And I think this is important. Generally, it can potentially be important to many other neurodegenerative diseases where you have a context where you have to really rely on this microglia effectively degrading the material. Let's also think about how the microglia are doing, you know?
You're really quite affectionate for the microglia.
Yeah, I think I watched them so much in videos. You know, I had to do so many quantifications that I became attached to them
it
emotionally.
like it,
but I guess it will be, yes, what about us, you know? Who thinks about our well-being?
Okay, maybe to finish, what advice would you give to a starting PhD researcher?
I will say be resilient. Be resilient.
Like the microglia.
Like the microglia. Yeah. Just be like the microglia. Because there's going to be challenges and it's impossible not to take it on a personal level, because if you're doing a PhD, you put your whole heart into it. You will build up resilience through time, but I think it's already good if you have this mentality, this thick skin.
And find something that can help you reacidify yourself, right?
Exactly, reacidify yourself.
Yeah.
We're going to make some stickers, bumper stickers. Yeah. Okay, thank you very much and good luck with your presentation this afternoon.
Thank you.
Thank you. Lovely to have you here.
Thanks.
Bye. With our next guest on the podcast, we take a deep dive into our brains. Professor Emmanuel Pasciuto is group leader for the Laboratory of Neuroimmunology at the VEB Center for Molecular Neurology. She researches the therapeutic potential of brain-resident T cells to reduce neuroinflammation. Welcome to the podcast, Professor.
Thank you.
Maybe a quite obvious first question, but what are T cells?
Well, T cells are a component of the adaptive immune system, together with B cell, for example, and they help us fight against infections and autoimmunity, for example.
Okay, and they're white blood cell, or?
They are white blood cells, yeah,
okay.
Indeed.
Okay, and before we go any further, it's maybe necessary to also tell us what regulatory T cells or Tregs or Tregs, how do you?
They are Tregs.
What are Tregs?
So Tregs are one subset of the T cells. So there are different subsets of T cells. So there are the CD8 and the CD4s. And amongst the CD4, you have effector T cells and regulatory T cells. So, and they have a very crucial difference. While the effector T cells are more known because they actually fight against infection and they are producing inflammatory cytokines, the regulatory T cells have the opposite function. So they actually stop inflammation, and they prevent our body to react against our own body. So they prevent also autoimmunity.
Okay.
So their role is to stop excessive immune response.
Okay. And where do they come from?
So they have the same origin. They also are selected in the thymus, like normal CD4 T cells, but they are specifically selected for their ability to bind self-antigen, for example. So they have simply a different type of selection, but they have exact same origin as other T cells.
Okay, and the thymus is...
The thymus is most prominent in the children because during the first period of birth is where we develop our mature immune cells, while the cells are selected for their ability of not reacting against our body, except Tregs. And after the thymus gets shrinked, so it's very much reduced in adulthood, but it's still there, it's just very small.
And it's still producing T cells?
So the T cells are actually produced in the bone marrow, and then they go through selection in the thymus. So yes, the T cells are produced throughout our life.
Okay.
Maybe to zoom out a little bit, why does our immune system sometimes overreact when staying calm would actually be better for our health?
So it's not actually like that. So the immune system needs to react, and that's essential. So, but this response needs to start and needs to stop. So, and this is a physiological response. And this is why, so the Tregs are there to allow the response to stop at a certain point. And if this does not happen, then it goes wrong.
Okay. And you talk about the complex interaction between the immune system and the nervous system. But aren't all interactions between the immune system and the nervous system incredibly complex?
Yeah, indeed. This is true. So, but... I think I found this especially complex because there are so few T cells in the brain. And I find so fascinating to see how just a few cells in one big tissue have such an impact. And this is because of specific property of the T cell. So I think it's a very specific challenge to understand as such rare cells can interact with so many neurons and influence them. So this is why I think it's especially complex.
And is it only are we just only beginning to understand how this system communicates?
Yeah, we really don't know much about it. So because so far we used, one challenge is that there are very little cells. So it's very challenging to study in detail their proteomic profile. So now we just limit our knowledge to transcriptomic because we have much more advances in single-cell transcriptomic. But I think now our challenge is to try to really understand which protein do they express, because they are the one that do the job. So they are the one that allow communication with other cells. So we are now moving towards more maybe genetic tools to study this interaction rather than relying just on single-cell transcriptomic.
And could it be that some modern diseases are linked to pathways between organs or systems that we haven't fully explored yet?
Yeah, for sure. So, I mean, one example is, for example, autism, where we really didn't know that there was a role of T cells. So, actually, when I started my PhD in 2009, so I was working in autism model, and yeah, we really didn't know that the adaptive immune system could be involved at all. And I because I was so fascinated already at the time about the T cells and the adaptive immune system, I started looking everywhere, and I was so surprised that it was not done. And now, like just maybe a few weeks ago, a clinical trial was released in autism for modulating Tregs. So I think this is really a new era. We are learning so many new things about the adaptive immune system and the brain.
Okay, and on that note, you write that you broke the dogma of the brain immune privilege. What is that?
So, well, for many years we thought that there were no immune cells infiltrating into the brain, and specifically not adaptive immune cells. So we knew that there were just these resident cells like the microglia in the brain parenchyma, but we thought that it was completely sealed off from anything else, that nothing inflammatory could enter into the brain. And this is, I have to say, even though there are, besides my study, more publication already showing that, this is still, I feel, a bit a dogma in the field. So I really would like to spread the word and get this accepted, so that actually T cells are in the brain and they do not necessarily trigger inflammation. We just need to understand them better and know what they are.
Okay. And the levels of the protein EL2 can limit the number of T cells in the brain. Can you explain how this works?
So, well, it's not exactly this way. So basically, IL-2 is a cytokine which induces proliferation of T cells in general through binding a receptor that is called CD25. So, and this is especially true for the Tregs that especially rely on this cytokine to proliferate and survive. So now in the brain, this cytokine is very low. It has a very low level. So this is why we believe. So we perform some specific experiments. They are called parabiosis experiments, in which two mice are bound together, and you can study the kinetic of the cells. So what we can do is, because each mouse has a specific genotype, you can trace the cell. So for example, imagine a cell is always called A and B. You can always recognize in any moment A and B, and you can trace how a cell from one mouse goes to the brain of another mouse, and how do they change each other. So, and we use this model to calculate over time the kinetic of the T cells in the brain, because we had a time course. And we could see that the Tregs would just die very quickly. So while the other subset of T cells would accumulate in one other brain, not the Tregs. So, and we think that they are going to die. And this is because there is not much IL-2 into the brain.
Okay.
So you had also, you did also research where you delivered IL-2 boosts inside the brain.
We have developed a specific strategy in which we use a virus which only expresses into the brain. So this virus can be injected IV, so it's not invasive at all, but can reach the brain only. And then we get IL-2 expressed either by astrocytes or by neurons. It works very well in both cases, and then IL-2 is released very low dose in the brain only, not outside.
And it has an impact on which diseases?
So we studied so far the impact on traumatic brain injury in mice, and then also in stroke, and in a mouse model of multiple sclerosis. And I'm currently studying the impact in Alzheimer's disease and autism.
Okay, and you had quite a positive result?
So for Alzheimer's, it's going quite well, but for autism, we just started, but I already know that there are also other groups working on it, and it seems very promising. So I'm very positive that it will work.
Okay. And yeah, we have to mention the Nobel Prize in Medicine, I think.
Yeah. I also mentioned it in my talk.
Okay. Because in the Nobel Prize in Medicine 2025 has recently awarded research in this area. Can you tell us a little bit more about that connection?
Yeah, well, so the Nobel Prize was given for the research in immune modulation, and I was very happy to see it. And there was one of the researcher that was received the award was Sakaguchi. So that, when I started my studies in immunology, it was every review I read about Tregs was from him, because he's the one that discovered actually the genes that is associated to this population that was defined as immunosuppressive, that are the Tregs. And then the other two researchers were the one that actually discovered the syndrome. So this is the IPEX, because actually absence of this Treg is very dramatic, and is actually lethal after a short time after birth. And this is called IPEX. This is where Tregs are missing, and the FOXP3 genes that is associated to the pathology is actually mutated.
Okay. Yeah. So you were a big fan?
Yes.
Yeah. Okay, that's great to hear.
Just one final question. What advice would you give to a young aspiring researcher or scientist?
Yeah. So, well, I think something that I would advise is that they always have to follow their ideas and don't give up, science is not easy, and answers are not simple. So this is a job that requires a lot of dedication and passion, and so they need to be really convinced and give themselves if they want to pursue. Otherwise, it's not going to work.
And find your own dogma to break, maybe.
Yes,
could also be.
Well, something that I would like to mention is that, is a bit generic. I'm really happy that I could continue these studies, because being able to link what I learned during my PhD on neurobiology and neuroscience to what I did during my postdoc in immunology is something I really wanted to do. And now research is very challenging. It's very tough to start a lab, and I'm so proud that I could do that here in Antwerp. And I found a very collaborative environment, a lot of input. I really hope you can hear more from us in the future.
Yeah,
so
I'm sure we will.
Yeah.
Okay, thank you very much, and good luck with all your research.
Yeah, thanks.
Thank you.
Our next guest is PhD researcher Arthur Steegmans. He's working at Naturapt, that's Natural Products and Food Research and Analysis, Pharmaceutical Technology at the University of Antwerp. Welcome, Arthur.
Thank you. Thank you very much.
Arthur investigates spray freeze drying as a novel drying process for the formulation of probiotic powders, and this with, and I'm going to do my very best to get this right, No, that's not right. Lacticacibacillus rhamnosus GG as a model probiotic strain.
You can say LGG if you want.
Yeah, but I just had to pronounce it once.
Yeah.
So, Arthur, let's start at the beginning. What are probiotics?
Probiotics are living bacteria which, when you administer it to a host, provides benefits to the host, benefits on health benefits on the host. They are already situated in the gut, in your mouth, on your skin. Microflora are everywhere, and probiotics are healthy bacteria to provide a health benefit.
Okay, and there's always a hint of pseudoscience surrounding probiotics. How are they applied in a clinical context?
In a clinical context, there are already several studies provided by the lab of my co-promoter, Sarah Lebeer, which are proven to have a benefit on the hosts. So I would not call it pseudoscience at all.
Okay.
Probiotics is actually a thing. It is already applied in the clinical context. There are a lot of probiotic formulations already available on the market, but not as a pharmaceutical preparation yet.
Okay.
How important is it then to have clear quality standards concerning these probiotics?
It is important because you want to be honest with your patient. And if you want to claim clinical benefits or health benefits, then it is important to also have the right quality standards applied to the product. That's for every pharmaceutical compound, live bacteria or dead compounds, chemical compounds.
Yeah, and you're working with live bacteria. I'm going to try it as well. The Lacticacibacillus rhamnosus, right? What were the reasons to choose this bacterium?
LGG. I'm not going to repeat the scientific name again, but LGG is one of the model strains probiotics already in use. I think there is already one commercial preparation available for children, actually, but it is a strain nice to use because it is more robust than wild types. It is a wild type, but a more robust wild type, and the proof of concept is nice to prove it on a model strain type of bacterium. Now my research work already involves other strains as well, but I didn't want to go in depth in my presentation. But
one.
I'm also using other strains as well.
Okay.
Wild types isolated from clinical, I would not say patients, volunteers. And I'm also implying this in my research work already.
Okay. And you have spray drying, freeze drying, and you combined them into spray freeze drying. What are the advantages of merging these two techniques?
The advantage is that you have the gentle drying process of a freeze drying process, which is a principle of sublimation process. But you have a loose powder in the end, which is a big advantage in spray drying. Freeze drying is the state-of-the-art technique currently used, but the milling process is required after freeze drying, because the end product of a conventional freeze drying process is actually a freeze dried cake rather than a loose powder. So that it's more processing steps before you have an end product. And that's the benefit of spray freeze drying. That is that you have everything combined, both of the advantages of spray drying and freeze drying in one process.
Okay. And when this technique is fully developed, how can it be applied?
It can be applied by developing products in a loose powders, just like powder bags, for example. Mm-hmm. But I'm now also testing in my research work compaction experiments with the loose powder. So I'm doing compaction experiments with the spray freeze-dried powder to see if compaction is possible with the probiotics in the dried state. And the results are very promising.
And compaction, what's compaction?
So we're producing tablets.
Okay.
Simple as that.
Okay.
We're producing tablets with a spray freeze-dried powder, which was not yet described in the literature before.
Okay.
So
you used the word tabletability in your presentation,
exactly,
right?
Yeah.
What are the important factors to take into account to make the perfect tablet?
The tablet should not break after a certain force is applied, but it should also not be too hard that it's not decomposed in the human body. Next big thing about tableting is the bacteria should survive the process. So that's actually
Preferably.
the main question about tableting is, do the bacteria survive the compaction process or not? So that are actually the two main goals in tabletability, is finding the right properties for the tablet and see if the bacteria survive the process.
And what's the benefit of making a tablet instead of just having the powder in a
capsule?
In a capsule, for example, or in like a bottle, or in, how do you call it, in a sachet?
Yeah, or a vial, for example.
Yeah.
Tableting is the most used production technique for oral dosage forms. Filling the powder in capsules is also possible, but it is a more critical process than tableting. Tableting is a very robust process. Most of the manufacturing sites in pharmaceutical industry are equipped with already tableting production lines.
Yeah.
So it would be nice to have a proof of concept designed for what is actually available in the industry.
Yeah. Okay.
And you also have the melt under your tongue tablets.
Yeah. These are also the lyophilized tablets, like Motilium, for example. These are actually tablets which are also produced with a freeze-drying technique. So they are freeze-dried already in the aluminum packaging.
Okay, that's why they look so funny. Yeah.
And that is also a very, very critical process.
Okay.
So we eliminate the criticalities of the freeze-drying, and we make a bulk product with the spray freeze-drying, which is suitable for multiple further processing techniques.
Okay.
Yeah.
Okay. Very interesting, yes.
Yeah.
So your spray freeze-drying has a lot of steps in the protocol. Eh? Was there a specific step you really had to fight to figure it out?
I did the experiments in a production site in Germany. So I got to this partner via my previous experience on my previous job. And we had the chance to try the process as deep as we wanted to. But the challenge was that the lab is here, where we grow the bacteria, and the production site is in Germany, which is almost 7 hours drive.
Not good for your bacteria.
Living bacteria, you can already imagine if you are if you're producing the bacteria and you do the drying process, the conditions are not ideal. And we already repeated several experiments now this summer, and we tried to tighten the timelines a little bit, and now we got even nicer results than which were published already earlier this year. But if we would have the production site here in Antwerp, then we could optimize the production.
So it's just a logistical problem.
Yeah.
Okay.
Because we work with living cells, and
yeah.
they like to be in the water, but in an aqueous environment, but not for too long, because then they die. And that's the reason why we dry it.
Yeah.
So
Okay.
Is there some other technique you use in your PhD?
Yeah, so I'm now focusing for this day on spray freeze drying because it's a relatively novel technique. But I'm also investigated spray drying and optimizing spray drying as another drying technique because we have a lab scale spray dryer at Campus Groenenborger, which is comparable to what is also equipped in several pilot scale manufacturing sites. So it is really nice to compare different drying techniques and see one-on-one what are the benefits and which technique is suitable for what end product we are looking for.
And it's closer than Germany.
That's a fact.
And for me, Arthur, your research sounds super technical. What especially interests you about the technical side of things?
Yes, interesting question. Thanks for that. Actually, I graduated in 2019, and in pharmaceutical care, and I started my career at Pfizer in Puurs in manufacturing environment, which is a little bit atypical if you study pharmaceutical care. But then I got involved in freeze-drying processes. I was product and process owner of several aseptic freeze-dried products. And then I got the opportunity to start a PhD here at the university. And it was really nice to have my experience on the field and put it in my research.
Yeah. Okay.
So that's, it kind of, evolved a little bit the technical interest, but in my hobbies I'm also kind of interested in technical things. Yeah.
Like
maintaining my bicycle. Maintaining a mountain bike, preparing the skis for the next winter. We are renovating the house. That's
almost the same as making pills.
Totally other skill sets, but complementary probably.
Okay, thank you, Arthur. Before we wrap up, one last question: what advice would you give to a young starting PhD researcher? Who doesn't have to be young, just a starting PhD researcher.
Yeah, because I was not young at all.
That's still young.
Sorry.
That's why
correction. Still young.
Why I rephrased my question.
Your research is what you make of it. So your imagination is the boundary of your research. And if you just try and see what comes next, then your research would also be a fun journey. If you wait for opportunities to come towards you, then I think the chances are smaller that something nice will happen to you. That's also, I would like to thank my supervisor for that, for not limiting my naivety as a young researcher and to see what is possible. The compaction experiments were actually not on the radar before, but when I was doing the analysis in another lab, they had a compaction simulator and they said, oh, maybe we should just try it and see where we get. And we did some preliminary experiments and they tend to be better than we expected. So now we're in the compaction story, which I did not expect starting my PhD at all. Yeah.
Okay. That sounds like an adventurous path.
It is. The journey is the destination.
Another bumper sticker.
Another bumper sticker. Every episode we have one bumper sticker.
Oh, nice.
Not really, but in this one we have another one.
Okay, Arthur, thank you for coming by.
Thank you, Arthur.
And good luck with your presentation this afternoon.
Thank you very much.
Our next guest is a PhD researcher and has been on the Faculty Focus podcast before, where she talked about the chemicals that end up in novel plant-based foods. Now, the title of her research presentation is even more scary. Chemical Exposure in Childhood, a study of possible exposure sources and health effects. Welcome, Fatima den Oude.
Thank you. Thank you.
Humans are daily exposed to a mixture of chemicals. Why are children especially vulnerable for exposure to those chemicals?
So there are two reasons for that. The first is that they are different from adults. They have different physical characteristics. Their metabolism is not yet completely developed, but also their body surface area is different. And also important is that they have different behavior. So they spend more times on the floor where dust can accumulate. So they are more in contact with dust. But also they have more hand-to-mouth contact because, of course, they touch everything, put everything in their mouth. So they are exposed already more. But also their body is still developing. And a lot of chemicals can affect development of organs. So then the children are especially vulnerable for that.
Okay.
Yeah, you already mentioned a few of the exposure routes. Are there other exposure routes?
Yes, diet is an important exposure route. Dust, as I say, but also children toys. Of course, they play with a lot of toys, and they can also contain a lot of chemicals. And for example, the flame retardants that I'm working with, they are known to be in foam. So the foam that's in your mattress, for example, but also the foam that you, I don't know exactly the English word.
In the baby bed.
Yes, the baby bed, also where you change the diapers.
Ah, yeah.
Oh.
That's a little better.
The diaper changing bed.
Yes.
Yeah, that
I
thing,
don't know
yeah.
the exact. But that's, so,
Okay.
we are really exposed daily.
Okay. You already mentioned some chemicals, but which chemicals do you target in your research?
So in my research, we focus on flame retardants. So as the name says, they are added to products to prevent fire and also to prevent fire from spreading if the fire is there. But they can even be used as plasticizers as well. And then I focus more on the classic plasticizers, so legacy phthalates and alternative plasticizers. And I also have done pesticides, but this is still in progress. So for that, I don't have results yet.
And do all these chemicals share common pathways?
PFRs and plasticizers, actually, they have quite some shared pathways because we see them in the diet in the same products. And also, as the flame retardants can be used as plasticizers, we see them also in plastic products. But plasticizers, for example, are also in toys, while flame retardants you will probably find less in toys.
But for example, for mattresses, it's like a little bit choosing between the pest and the cholera. Do we want chemical exposure or do we want a flammable mattress?
Exactly. So that's, I always say, okay, I look for these things, but I also don't want to cause panic because also with pesticides, they are quite useful. And indeed, flame retardants, if we would not have them, if your couch goes on fire, then your whole house goes on fire. Yeah. So we need them as well to live a safe life.
Yeah.
So it's always a balance.
And for your research, you used samples from the Flemish environage cohort. What's that?
So this is a cohort from Limburg. There is now currently 2,400 mother-child pairs involved. So they are still being recruited. So it's a prospective cohort. And then when they recruited them, they follow them throughout their lifetime. So I think the oldest ones are now around 14. So they do multiple measurements, and we have used samples from the measurement period 4 to 6 and 9 to 12 years old.
And those are urine samples or blood samples or?
I focus on urine samples for the biomonitoring, but we also have blood and feces, and that we use, for example, to check inflammation. But there's also questionnaires that the parents fill in, and they are really useful for the exposure sources determination. So they donate a lot of samples, even saliva, the placenta, that I don't use, but other people in my project use them.
You also mentioned that having a pet in the house increases exposure to the chemicals you study. How so?
So we found a link is too strong because I think it's only associations that we can determine with regression. It's not a causal linkage. It's an association. We found that children with a pet had higher exposure to legacy phthalates. But we should interpret a bit with caution because the sample size is not super high. But we think it might be that, of course, the pets, if they have hairs, they collect the dust in their hairs. And if children who like, of course, pets, they touch them and then they go with their hands in their mouth again. And then they can be exposed through that route. And we don't know why we only found it for these kind of compounds, but maybe here it's more prone to accumulate in the hairs or something like that.
Okay, when I read that pets could be one of the exposure routes, I immediately thought, probably like the anti-flea necklace, but that's not the chemical compounds you're looking for.
That would be more indeed for pesticides. Here it's we think it's related to dust, but we did not find any other studies who even looked into this association. So we can also not say, this is in line with what we saw. We are also now in new studies where we are involved trying to say, if you have a questionnaire, please include pets. Because, you want some validation in the other cohorts to see if it's really like this or just a chance finding.
You already mentioned the term legacy phthalates. Does the term the legacy refer to pollution we've inherited, or where does it refer to?
It's more legacy in the sense of classic, that they were first on the market. And for those phthalates, it's now quite clear that they can be endocrine disrupting. So in the EU, but even I think in the US, there are now some restrictions and bans. But what happens when you ban something? A new thing will pop up, and that are the alternative plasticizers, and I think that's why we call them legacy, to make the distinction between the classic ones and the alternative newer ones.
Are we also creating new chemical legacies today for the future generations?
Oh, that's difficult to answer, because that's what we are trying to see also in our study, that those newer compounds, we don't know yet much about their health effects. So that's also why we say that they should be monitored more, because we can see that the legacy phthalates, they are decreasing. When you compare concentrations, what we found with previously done studies, you clearly see a decrease, and this is a trend worldwide. And this is good because it shows regulation works. But on the same time, we see those alternatives increasing. So we also say, as a recommendation, keep monitoring them and keep researching them, because the toxicity tests that have been done show that they are less toxic. But of course, you never know in real humans, so it should be monitored. But we cannot say clearly now, in 10 years it's really bad. So that's a bit why we chose those class to say, we need more information. Look, we find them.
And you make a link between chemical exposure and the alternations in the gut microbial diversity. Can you elaborate on that?
Yes, so in our cohort, we have access to a lot of data because it's a collaboration between different institutes. So we all measure different things. And in one institute, they measured the gut microbiome. And the gut microbiome is very important because it's responsible for a lot more function than people think. They help with metabolism, with your immune system. And we did look for associations between concentrations of the compounds and the gut microbiome. But actually, I showed in my presentation that we only found for one metabolite that it can have effects on the gut microbiome. It showed a lower richness. But I have to say these results are still quite preliminary. That I've only done analysis a few weeks ago, so we still have to see. But for now, actually, it seems that for most of the compounds, it did not have an effect on the gut microbiome. We only found one that a decreased richness was seen.
Okay. That's
Good news.
That's good news.
Yes. I'm not here to create panic. I'm not here to create panic. No worries.
So you mentioned already the data analysis. You collected a lot of samples and then took on quite some data analysis. And in your presentation, you briefly mentioned the challenges that come when analyzing data of big data sets. Can you tell us a bit more about what you can encounter when dealing with big data?
So it's a cohort study, and in the beginning I thought, oh, nice, then you have all these nice linked data. But I also learned now that it's very difficult because, we are multiple institutes, but then we did not all get the same sample, so from the same person. So already you decrease your sample size if you would use only the overlapping samples. And then also the questionnaires that in the beginning I thought, how nice. But then you receive them and a lot is not filled in, and I got very upset. But then when I saw the questionnaire, I understood the people because it's really huge, really, really, really huge. Like I was invited for a questionnaire myself that took an hour, and I was already thinking, too long. But this one I think will take a full-on morning or afternoon. It's really big. So I understand the people. But of course, it decreases your statistical power because you don't have all the data. Then you have to think about which covariates to include, and you encounter different problems that maybe your urine and feces are not taken at the same time point. So you have to do a lot of thinking and to think what are the best choices to make. And that's why I say it's a bit preliminary still, the statistical results, because we are still trying to figure out the best way.
So hooray for the Flemish environment cohort.
Oh, that's for, yeah. Well, I mean, yes, I'm very happy to collaborate in that.
Is there anything else you want to add to our talk that you think our listeners might find interesting?
Yes, I want to put a bit more maybe in perspective that it might sound all scary, as you say, but we also did risk assessment, and then you compare the levels that you find with levels that are thought to be a risk. And actually we found only few children to be at levels higher than the threshold, only 2 for legacy phthalates and 10 for alternative plasticizers. So if you take into account, I analyzed 600 samples. That's is quite an okay finding, I think.
And were there then some common factors between those 10 and those 2 that you could say, okay, they were exposed via this route?
No, indeed, one of my co-authors asked it when I prepared the manuscript, so I went back to all the questionnaires to see, did they have 10 pets or
for instance, or a cat and a dog?
I could not immediately see, did they consume this food group like crazy? But that I could not immediately.
They were eating novel plant-based foods all day long.
Who knows? Only there's one phthalate, DEP, and this is used in personal care products. So there you will also find a huge range. So you have very low and very high concentrations. And I think it's due to cosmetic use. But this was not included in the questionnaire. But there we are quite sure that these compound concentrations are very dependent on the personal care products a person use.
And you can't really address those subjects and ask, do you use those?
No, they prefer not to because of the anonymity.
Probably. I can imagine.
Maybe if you communicate back the results, if they want that, then you can say, if you want, we can discuss. Mm-hmm. But I think for now in the project, it's not the idea to really...
so many variables.
Mm-hmm. As I said, they already gave so much time to us.
That's true.
Yeah.
In the beginning, I thought, so easier go, but now I think, wow, those people voluntarily give a lot to us. Actually, it's really nice.
Okay, before we wrap up, we want to ask you one last question. Mm-hmm. What advice would you give to a starting PhD researcher?
Oh, that's a difficult question. I would say that's. In my second and third year, I had it quite difficult because you're working, of course, a lot. You do a lot of analysis, but maybe you do not see the results yet. Mm-hmm. So I felt, okay, I'm here with this instrument all day long. I come in the weekend, but I have nothing. But now that I'm finishing up, I can see, okay, I actually done a lot and it will come to a nice ending and you can combine your results. And so don't lose hope if it feels like you're doing everything for nothing. At the end, you will be there.
That's something we hear a lot, that the second and third year are the
most
hardest.
Yeah, first you're learning, all is nice, all is good. Then you start your work and you're just working without seeing maybe the results, but that's what I would like to say.
And then some technical
results
issues
will be there, and that's what I want to say.
Okay. Thank you very much for coming by, Fatima. And good luck with the rest of your PhD.
Thank you.
Next on the podcast, we're going to talk about natural products for human health with Assistant Professor Emmy Twenter of Naturapt. That's Natural Products and Food Research and Analysis, Pharmaceutical Technology. Welcome, Professor.
Thank you.
On your LinkedIn page, you write, and I quote, "My research is focused on phytochemistry, biotransformation, and bioactivity of natural products, exploring the health-promoting properties of medicinal plants, marine organisms, and food production in order to use them in the most safe and effective way is the central objective of the research projects I'm involved in. When it comes to food and health, there's a thin line between science and pseudoscience. Food research can be a difficult research topic, and why did you choose this path?"
Well, my background lies more in the pharmaceutical approach, so the medicinal products used as drugs. And it was during my postdoc where we did research on cocoa and chocolate, where I saw that there is actually a big overlap between foods and natural products used as medicine. And my idea is that the techniques that we apply for analyzing these natural products that are being used as medicine can just as well be used to improve the analysis of food products. So that's why now I'm broadening the spectrum, let's say, to also include these food products, but still focusing on foods that are beneficial for our health.
Okay.
So it is a very noble mission, helping to develop and implement appropriate methods as control mechanisms for plant-based consumables. Why is it important that such control mechanisms exist?
Well, what we see nowadays, products that are on the market, there are different types. We have herbal medicinal products, so these are actually registered as medicine. We have food supplements, which also can be based on natural products, so plant constituents. And you also have regular foods based on, let's say, plants or other natural constituents. And there are different criteria applying to the quality control. So safety always has to be guaranteed. That's very important, that consumers only can purchase safe products. But for the quality control, the regulations vary. And I actually think it's a shame. Many consumers are not aware, especially of the difference between herbal medicinal products and food supplements. And the control of quality on food supplements at this moment, in my opinion, is not as high quality as it should be. So there are food supplements, and we've contributed to some work, some reports by Testaankoop, for example, analyzing food supplements on their content of active constituents, and you find a very big variety. Some indeed contain what is stated on the label, and some contain much less. And I think that's something that it shouldn't be possible that this is happening in our country.
Yeah.
So
a lot of work in that field, I can imagine. Yeah. Did you hear about the Droomsap story?
No, I don't think so.
It's something in the Netherlands and in Belgium. It's some juice you can take to sleep better, but there are now a lot of negative health effects.
I think it's a mixture of different plants, I believe.
Yeah.
I read about it indeed.
Yeah.
And they're trying to limit the sales.
The side effects are really quite heavy side effects.
I think indeed it's a fair point. Many people think, it's a natural product, it will be safe, but it's not automatically that it's natural, that it's also safe to use. It can also have side effects. It can have interactions with actual medicine.
Yeah.
So cautious use is important.
Yeah. And there are so many variables when it comes to plants, right? What is the most significant challenge as a scientist working with plant-based materials?
There are many challenges, I would say. Indeed, you have batch-to-batch variations when you work with plant material, so seasonal variation, geographical variation. You also want to work in a sustainable way, so there are many things to take into account. And I think we're trying to improve or work on all these parts by, for example, not using roots anymore, but replacing these by working with leaves. Of course, you need to check that they have the same activity, but in that way you don't have to remove the whole plant, but you can just take a part and the plant itself can still continue to grow. And with regard to the variation, what is ideally done is that you perform a standardization of extracts, so you always quantify the active constituents and you try to have stable levels of these from batch to batch. But this is surely not done for all products on the market yet, so
a lot to be done.
Okay.
Yes.
And for centuries, mankind has used natural products to maintain health or treat illness. How do you decide which of these traditional remedies is worth studying in the lab today?
Well, what we do is sometimes we consult traditional healers.
Okay.
So this is not a common practice here in the Western world, but for example, in Africa or South America, this is really normal that people, when they have issues with their health, they consult a traditional healer and he or she then recommends a certain plant to be used. And so we also ask these people, okay, which plants do you recommend to which types of disease conditions? Mm-hmm. And then we do a literature study on the plants that they mention. We also collect the plant material, and especially the plants that are not studied a lot yet are of our interest. And then we do a first screening on bioactivity to see if there is potential, and if so, then we continue to look further into the active constituents.
Okay. And are you scared that all the knowledge that lives with these traditional healers is disappearing? Or is it important to write all that knowledge down?
Yeah, on the one hand, well, there are some communities where that's being done, like traditional Chinese medicine. It's well documented, but there are also other types of traditional medicine where it's just mouth-to-mouth communication, and the information goes from one generation to the next. And I don't think that will disappear, actually. It's still something very actively being done in these communities, and I think that will continue to be done in this way. I just think that the scientific data we have to support this use hopefully will only be extended, and we will continue to increase having the scientific data to support this use.
And when you scientifically prove that a certain plant that grows in South America has a lot of health benefits, is there like a danger that pharmaceutical companies start exploiting the land and taking out the plants?
Well, nowadays there is also a lot of attention for the benefit sharing. Indeed, in case a promising plant is identified, already from the start of the research, the Nagoya Protocol has to be followed. And so there is an agreement between the community providing the plant material and the researchers working on it to make sure that the people who originally were working with these plants also have benefits if a promising compound is identified. So that's taken care of. With regard to over-harvesting, that's not particularly included in the Nagoya Protocol, but I hope that the pharmaceutical companies realize that they have a responsibility to take, and they will not follow that kind of practices.
Yeah. They might grow their own.
Yes, exactly.
Yeah, or they can figure it out how to.
Like with the thalassia.
Indeed. Yeah. And sometimes there is also semi-synthesis, so you start with a natural product and there are some chemical modifications done later on in the lab that can also be done.
Mm. Yeah. And there's still a lot of things to learn about the marine organisms. But is there a part of your research that you believe holds the most promise or could have the biggest impact?
The project on sea cucumbers that is going on right now, I think there is really a huge knowledge gap there. And so there was a literature review we did a few years ago, and we saw that there were quite some chemical analysis done, but in a bit superficial way is not the right word, but screening in the extract, while actually it would have been necessary to go through the whole process of isolation to know which compounds are inside these sea cucumbers. And there is still a gap between this monitoring in the organism as such and the actual constituents. And of course, we first have to clarify this before we can go to the bioactivity of these constituents. So I think I'm very curious to see what will come out of this project. Yeah.
And also our gut is of more and more interest to researchers and seem to have more to say about our health than we might expect it 20 years ago. Do you think that influencing how plant compounds are absorbed and transformed in our body could lead to new treatments now or in the future?
I think it will take some time before that's the case. but there is a very interesting interplay between the microbiome, the gut microbiome, and these natural products. So on the one hand, gut microbiome is responsible for biotransformation, but we also see that some natural products like polyphenols can affect the composition of our gut microbiome, and they can have even like a prebiotic effect. So I think in that way we can also maybe improve or adapt our gut microbiome by consuming the right foods. So it's a complex system, for sure more to study there, but I think there's a lot of potential, and let's see what happens in 10 or 20 years, yes.
Good.
And for your research, you use an in vitro gastrointestinal model. Can you tell us how that works?
Yeah, so it is a model that mimics the gastrointestinal tract. So there is a stomach phase, a small intestinal phase, and a colon phase. And this last phase, the experiments are done in an anaerobic chamber, so without oxygen, like the actual state of our gut in our body.
Yeah.
And in this last phase, we have compounds or extracts of interest. We submit it to the different phases, and in the last phase, they are co-incubated with human stool samples to see the biotransformation going on in the colon. And at each stage, we take samples that are then being analyzed to see the different steps in the biotransformation.
Do you think that that's an important step for quantification, using such a model to guarantee the quality and safety of herbal products?
I don't think that's really important for quality and safety, but more for the efficacy, actually.
Okay.
Because the compounds that are responsible for the activity are the ones which reach our circulation, our blood circulation, and very often they are not the same as the ones that are present in the product we consume.
Okay.
So there is this link between the products we take in, then the biotransformation, and then the metabolites, which eventually will express the bioactivity.
So the plant itself is not always the medicine, it's what our body creates afterwards.
Yeah, it can be considered like a prodrug maybe.
Yeah.
So it's not the case for all natural products, but in many cases we see that that's the case. Yeah.
Okay. Before we wrap up, is there something you want to add to the conversation that you think that we missed and you think our listeners might find interesting? Probably a lot, but...
Well, I think, and it was also the main message I tried to convey during my presentation, when we teach students about analytical methods, it seems quite theoretical, perhaps even a bit boring. But I think when you see the applications and the goals that you can reach by applying these methods, that it becomes clear that they are really important and they are very useful, actually.
Yeah. Okay.
Good message.
Good message. And then I want to ask you one last question. What advice would you give to a young aspiring scientist?
Search for a topic to research or to carry out your research on that you really enjoy. I think that's the most important part. If you're motivated to perform your research, then you will get there in the end.
Okay.
So enthusiasm, that's
Check.
key.
Check.
Yeah.
Okay, thank you very much
Thank you,
to
Ruby.
come by, and good luck with your research.
Thank you.
And just like that, we welcome our last guest of this year's podcast marathon. Welcome, Sarah.
Hi.
Sarah de Beukelaer is a PhD researcher in biomedical sciences at the Laboratory of Cell Biology and Histology. She researches the drivers of cell state plasticity in glioblastoma with image-based contextual cell profiling. Glioblastoma, or brain cancer, why is there still no cure?
Mainly because the main reason that people still relapse is that there is like a small subset of cancer cells called GSCs, and they're very difficult to target. There's this cellular plasticity that causes the cells to constantly switch phenotype, and this is what makes them so hard to target.
Okay, and why is this type of cancer so aggressive?
Because it grows very fast, and also those GSC cells, they tend to invade strongly into the brain parenchyma, making it very difficult to resect.
Okay.
And you focus on cell state plasticity. Can you tell us more about this?
These GSEs, they have been defined using transcriptomic studies to contain 4 main cell states. So we have astrocyte-like cells, mesenchymal-like cells, oligodendrocyte-like cells, and neural progenitor-like cells. And those 4 main states, they have specific properties. They can also interplay with the brain parenchyma, the resident cells of the brain itself. And they invite strongly, which makes them very hard to target.
So to find these cells, you use a technique called cell painting.
Yes.
Tell us more about that.
So cell painting is a very easy technique. It's also developed to make our lives easier. Basically, it's leveraging one specific aspect of cells or cell identity that is often overlooked. Because when we look at those cells, we can also see that a neuron looks differently from an astrocyte, for example. Yet if we want to identify neurons and astrocytes in co-culture, we're still sticking to the methods that we know, using immunocytochemistry, for example, targeting specific proteins. But we can also use this morphology and use this to our advantage. And the cell painting approach stains not specific antibodies, not specific epitopes, but stains the morphology. And then we can combine information from the morphology with information from immunocytochemistry to train artificial intelligence, convolutional neural networks that are capable of predicting certain patterns and linking those morphological patterns to the cell state. And thus, in the future, making it possible to just look at the cell morphology and then output automatically what the cell phenotype or cell identity actually is.
And what do you use to give a color to the morphology?
The classical cell painting approach is based on some very simple dyes. So there's Höchstdordapi just to stain the nucleus, some concanavalin, which is an ER marker, wheat germ agglutinin staining lectins, also the cell membrane, the Golgi apparatus, for example, mitotracker staining mitochondria, and the actin cytoskeleton is also marked. So these are just really generic structures that are present within every cell.
Okay. We can all be moved by a beautiful painting. When will you feel satisfied with the picture of your glioblastoma cells?
When I think a picture is beautiful, personally, I always like to look at the nucleus. It's also something that is, a lot of people within our lab are really enjoying to look at a beautiful nucleus. Also, the actin, so the actin cytoskeleton often really gives beautiful images because it's like this entire network within one cell. It's really beautiful to look at.
So those are the 2 things you focus on, or you see?
No, this is just the looks.
When you look at it, you say, a nice nucleus, a nice...
that's nice, that I would hang in my living room.
Well, why not, right? I think there's something marketable about this.
Yeah.
You also developed a method to position and visualize assembloids in flow as an alternative to traditional in toto imaging. What exactly are assembloids?
Well, assembloid is derived from the word assembly and also organoid. So if we're looking at thinking about organoids, then these are iPSC-derived models. So we start from a stem cell, we develop them towards NPCs in 3D culture. And then this 3D culture, we develop it further in vitro to resemble the human brain. So this is like a small brain in a dish, if you put it simple. And then we assemble it with something else. In my case, for my assembloids, or to study the GSC invasion, it's specifically with a small tumor. So there I take the patient-derived GSCs, make them into small 3-dimensional structures, small spheres, so the tumors. And by putting those together, they start to fuse. They do this automatically without me having to do anything, basically, because it's the nature of those GSCs to invade into this brain-like structure.
Okay, and how do you image them? Or how do you get a picture?
There's multiple ways to study these 3-dimensional structures. So they're approximately 1.5 millimeters in size. So they're somewhat too big for the light to get through. If we're just looking at it by eye, it's just a white blob. So one option would be to cut them, to make slices of it, so that they become smaller. We can easily stain them and use regular microscopy techniques to visualize what's inside of our sample. There's one difficulty, certainly for these assembloid structures, where we're looking at invasion of these GSC cells inside the structure. We don't know where they are, and I do really want to gain a complete view of where exactly, so the spatial location of these GSCs in my sample is really important. So therefore, I do not want to cut the sample, because then I lose a lot of spatial information. So to make it easier, or to retain this three-dimensional nature of my samples, I'm making use of tissue clearing technique, where I make the sample transparent so I can completely see through it, and the light can also, the light that's used by the microscope, can pass through the sample and also reach the detector. So the sample becomes transparent, and then I also use light sheet microscopy, which is a more light-efficient way to visualize these millimeter-sized structures.
I see an enlightened brain.
And you describe in your abstract, you describe the classical approach of imaging as tedious. Why? Can you talk about a little bit why you use the adjective tedious?
I believe in that case I'm referring to the cyclic immunofluorescence approach, because there's a lot of the immunostaining relies on antibodies, and antibodies do not always work. They depend on the sample preparation, different conditions, also the supplier, differences from batch to batch. So this is something that's not always foolproof. It's also quite expensive, and I can only multiplex a certain number of channels. So I only have actually 3 slots available to combine different markers. But I have 4 subtypes, so I at least have to do 1 cycle of iterative staining to get a complete overview of what is exactly in my culture for those 4 subtypes. So that's why it's a bit tedious. I have to do a lot of work.
Okay.
It sounds like a good word for the process. Yeah. So I read in your presentation, or I saw your presentation up front, and I saw that you want to combine 2 approaches to gain better understanding of patient-specific differences. Can you tell us a bit more about these approaches and why you think combining them will be a good thing?
During my PhD, I developed those 2 approaches, the ones that we already talked about, so cell painting and also the high-throughput light sheet microscopy approach. So the first, I want to apply to have a better patient stratification. So I talked about the heterogeneity within the GSC cell cultures. And this heterogeneity and also the plasticity, so cells being able to switch from one state to the other, is said to be linked to the invasion potential and the aggressivity, or the aggressiveness of your GSC culture. So being able to get a better understanding for one specific patient, if a patient is coming from the hospital, and this patient has a certain pattern or certain presence of specific subtypes, we can get a very easy and broad view of which subtypes are already present within this patient using this cell painting approach. So this on the one hand, and then on the other hand, we have this high throughput assessment of cell invasion. So the first approach was cell painting, so it's static, but it's just the different subtypes that are present within the patient. But we also want to see how these GSC cells are behaving or how they are invading within the human brain. So then we get also a behavioral readout. And I believe by combining both, we can really associate the subtype presence with how the cells are invading, and in that way get a better understanding of how certain treatments might work for the patient, but also maybe give a better idea on the prognosis, see how this patient is going to evolve, which drugs might eventually work, and also in further drug development pipelines include all this patient variability to find novel drugs that can work on different types of patients.
Yeah, but the first application would also be individualized medicine, maybe.
Yeah, exactly.
We'll wrap up with one last question. What advice would you give a starting PhD researcher?
Fuck around and find out.
Okay.
A last bumper sticker
That's a good one.
of the day.
Okay, great. Thank you very much, Sarah, for coming by.
Yes.
And good luck with your PhD.
Thank you for the invitation.
Thank you, Sarah.
Okay, so that's a wrap for this year's podcast marathon live from our Faculty Research Day. A big thank you to all the professors and PhD researchers who took the time to join us and share their work. We learned so much today. And thank you, Marleen, for being a wonderful host once again.
Thank you as well, Teun.
And of course, we'll be keeping a close eye on all the research happening within the Faculty of Pharmaceutical, Biomedical and Veterinary Sciences at the University of Antwerp. So make sure to subscribe to the Faculty Focus podcast so you won't miss a thing. Bye.
Bye-bye.